Hello everyone, my name is Suyash Jagtap and I am M Tech student specializing in food technology at the MIT School of Food Technology, MIT ADT University. Today I am excited to present on the topic that is highly relevant in food industry, the adverse effect of genetically modified food on human health. In this presentation, I will cover a brief history and introduction of genetically modified foods, the potential health risks associated with their consumption, environmental and socio-economic impacts, and finally, a look at the global regulations and future direction in the field of GMO. Through this presentation, I hope to shed light on the both benefits and potential dangers of genetically modified food, giving us a balanced perspective on this important issue. Let's begin. So, the outline of the topics that will be covered are first, introduction to GM food. Second is health risk. Third, environmental and socio-economic impacts. Fourth, regulation and safety. And fifth, case studies and future direction. So, in introduction, in introduction, we will discuss the overview of genetically modified food, which explains the what are the GM food, how they are made, and types of organisms that are commonly modified. For example, corns and soybeans, why GM food are created, and the science behind GM technology and genetic engineering. So, the next is the health risk. In the health risk, we are discussed potential health hazard, toxic in that toxicity, allergy, cancer risk, and antibiotic resistance. Then environmental and socio-economic impacts. In this section, we will discuss about the environmental consequences of cultivating GM crops. There may be ecosystem disruption, biodiversity loss, impact on soil health. Then socio-economic impacts, for example, farmer dependence, cost of genetically modified seeds, global market dynamics. Then next is the regulation and safety. In this part, we will cover the regulatory framework that govern the production, distribution, and consumption of genetically modified food. Then last is case studies and future direction. In this section, we will discuss the real-world example of genetically modified food, highlighting the case studies of success and controversies. These may include the successful GM crops and controversial cases. In future direction, the role of newer technologies like CRISPR making gene editing more precisely. Okay, so let's begin. In introductory section that defines what genetically modified foods are and provide the rationale behind their creation. So the definition of GM food is the genetically modified food are described as a food that comes from organism whose genetic material means DNA has been artificially altered in the way that do not naturally occur. The modification is done through genetic engineering, which allows scientists to change the DNA of crops or animal to introduce new traits. Then what is the purpose of GM food? In that, to enhance certain desirable traits in the organism. These traits are typically aimed at addressing challenges in agriculture and food production, such as increased resistance to herbicide and improved nutritional content. Okay, then genetic engineering. So this refers to the scientific process of modifying an organism genome by adding, deleting, or altering specific genes. Scientists can create plants or animals with new beneficial traits. The resulting genetically modified organisms are then used to produce the GM food. It shows the development of genetically modified food in the scientific discovery and technological development. Okay, it highlights the scientific significant milestones in the history of genetic engineering and the creation of GM crops. In, for example, 1944, there is a DNA transfer discovery. This year marks a pivotal moment when scientists discovered that DNA is a hereditary material responsible for passing genetic information from one organism to another organism. This finding was fundamental because it revealed that genetic traits could be transferred between organisms. So, the discovery laid the groundwork for future experiment in genetic manipulation as scientists now understood that DNA held the instruction for building and maintaining all living organisms. Also in 1963, there is a genetic code deciphered. In that, the set of rules by which the information in DNA is translated into proteins was deciphered in 1960. So this breakthrough allows scientists to understand how specific sequence of DNA determine the structure and function of protein which are essential for life. Knowing how genes translate into protein gave researchers the ability to start manipulating these processes. By changing or inserting new genes, they could alter the traits of organisms. In 1973, there is a recombination technology of DNA. In that, the development of recombinant DNA technology in 1973 was a crucial advance. So the scientists learned how to combine DNA from different organisms, effectively creating genetically modified organisms. This process, known as gene splicing, allows scientists to cut and paste genes from one organism into another. The technology made it possible to introduce specific traits such as pest resistance into plant by transferring foreign genes. Then last is 1944, FDA approves the first GM crop. This marks the first time that genetically modified crop was approved for commercial sale by US Food and Drug Administration FDA. The approval was given for the Fleur tomato, which was engineered have a long shelf life by delaying its ripening process. The approval for the Slur tomato was a major milestone in the acceptance of GM crops for human consumption and paved the way for widespread cultivation and sale of GM crops around the world. So, here are some common examples of genetically modified food: corn, soya, cotton, papaya, and rice. So, the GM corn is one of the most widely grown crop globally. It is engineered for traits like pest resistance and herbicide tolerance. In soya, the GM soybeans are commonly used in processed food and animal feed. These soybeans are modified for herbicide resistance, improving yield and reducing weed control cost. The cotton has been genetically engineered to be resistant to pest, which has significantly reduced the need for chemical insecticides. And papaya, in that, the papaya was developed to resist the papaya ring spot virus, a disease that devastated papaya production in 1990s. This is the example highlights the role of GMO in saving specific crop from extinction. And also the rice. The golden rice is the best example of GM crop developed to address a specific nutritional deficiency. It is vitamin A. So, why did humans start producing GMO? The reasons for producing the GMO is increased crop yield. So how one of the primary goal of creating GMO is to improve crop productivity. By introducing genes that provide resistance to pest, disease and environmental stresses like drought, GMO can increase the efficiency of food production. So this helps to address the global food demand, especially in regions with the high population growth or unfavorable farming conditions. Then pest resistance. Crops are engineered to be resistant to pest, reducing the need for chemical pesticide. This is achieved by inserting genes that allow plants to produce substances toxic to certain pests. For example, Bacillus thuringiensis Bt genes, and which provides a protein toxic to insects. Pest-resistant GMO can reduce crop losses and lower the environmental impact of agriculture. There is also herbicide tolerance. This is the another major motivation is allow crop to tolerate herbicide, enabling farmer to apply herbicides to control weeds without harming the crops. For example, crops like GM soybeans, means genetically modified soybeans, have been modified to tolerate glyphosate, a common herbicide. This innovation reduces labor and chemical use in farming, enhancing its efficiency. There is also the improving the nutritional content. Genetically modification can enhance the nutritional value of crops, such as adding vitamins and minerals. A notable example is golden rice, which is engineered to produce beta-carotene, that is the precursor to vitamin A, to address vitamin A deficiency in some developing countries. And there is also drought tolerance. In region facing water shortage, drought-resistant crops can thrive in arid conditions, helping to ensure food security in those areas. This explains the three major methods used in genetic modification to produce genetically modified crops. So, the genetic modification method first is gene introduction, plant regeneration, and gene expression optimization. So, in the gene introduction, this involves the inserting new genetic material, a desired gene, into organism DNA. The gene introduced could provide traits like pest resistance or improve nutritional content. In that plant regeneration, after introducing new gene, plants are regenerated from single modified cells. This process involves growing a new plant from these genetically modified cells in a lab, which is then propagated. Then next is gene expression optimization. Once the new gene is inserted, it is essential to optimize how it is expressed. This means ensuring that the gene is active in the right part of the plant and at the correct level to achieve the desired traits. There are three major techniques to modify the gene. First one is directly transfer DNA, second was indirectly using bacterial vehicle, and third, direct editing of genomic DNA. In that, the directly transfer DNA, in this method, DNA is directly inserted into the organism genome using physical method like gene guns or chemical methods. These techniques allow for the introduction of new traits, which is developed in 1980s, also known as the microparticle bombardment technique. And using a bacterial vehicle, Agrobacterium tumefaciens is a type of bacterium that is naturally transfer DNA to the plants. Scientists use this bacterium as a vehicle to introduce foreign gene into the plants. This method is widely used for creating GM crops like tomatoes, cotton, and tobacco. It is also known as designer genes. Then next one is the direct genome editing, CRISPR CIS9. It is a revolutionary technique that allows scientists to make precise edits to an organism's DNA. Instead of introducing foreign genes, CRISPR can directly edit the organism's existing genes, allowing for more targeted modification, which is developed in 2012. CRISPR has been a game changer in genetic engineering due to its precision, efficiency, and lower cost. In this video, we see the what is the genetic modification. So, the natural selection tends to the favor plants that can compete for light, water, and nutrients, defend themselves from being eaten, and disperse their seeds over long distance. These characteristics are in direct conflict with the goals of agriculture, where farmer wants to plant to invest as many of their resources as possible into making nutritious, easy to harvest product. Genetic improvement has been a central pillar of improved agriculture productivity for thousands of years, with the modern crop varieties being much higher yielding and generally more nutritious than their wild ancestor. Even with our modern crop varieties, continued improvement is both possible and necessary to meet the growing demand. The new characteristic can be introduced into the crops using either conventional crossbreeding or a genetic modification, that is GM for short. The characteristic of living organisms such as size, color, and shape are determined by their genes and their interaction with the environment. With the improvement in our knowledge about which plants gene do what, we now know many genes that could contribute to improving sustainable food production. In some cases, conventional breeding will be the best way to deploy these genes, that is by crossbreeding the crop variety with the plant that contains the genes providing this characteristic. In other cases, genetically modified where scientists take a genes and insert it directly into the crops might be easier or indeed the only way they can be deployed. So this is all about the genetic modification. So up to this we see the what is the genetically modified food or how it works and what are they. So now we are discuss about the potential risk of genetically modified food by focusing on the three key areas, that is the human health, then ecosystem and biodiversity, and then hazards. So, in the human health, we concern about the long-term effects of GM food on human health, including allergies, antibiotic resistance, and potential toxicity. And then ecosystem and biodiversity, the impact on the environment of GM crops, such as the harm to non-targeted species, gene flow to wild population, and loss of biodiversity. In hazards, there is a possible unintended consequences like the creation of new allergens or toxins in food, or resistance in pests and weeds. In short, it stresses the possible health, environmental, and safety risks associated with GM food consumption and production. Effect of GM food on human health. Genetically modified food have been a subject of global debate, with various studies evaluating their potential effects on human health. Proponents of GM food argue that they are safe for food consumption, citing regulatory approvals and risk assessment. However, concern persists regarding long-term health effects. So let's explore the key concern. The first one is allergy. The concern is allergenic proteins. One of the primary concerns surrounding GM food is the potential for allergenic reaction. Genetic modification involves transferring gene from one organism to another. If gene from a non-allergic organism is introduced into a crop, it could cause an allergic reaction in people who are sensitive to that allergen. For example, if a gene from nut, which is a non-allergen, is inserted into another food like soy, individual allergic to nuts might experience a reaction when consuming GM soy products. There may be new allergens. The process of inserting foreign genes may create a new allergen that were not previously present in the original organism. Since these allergens are novel, the human body may react unpredictably. Then the next one is the toxicity. The concern is unintended toxins. Genetic modification could result in unintended biochemical changes in the plants, potentially leading to the production of harmful compounds or toxins. This could have a harmful effect when consumed by humans over a long period. When genes are inserted into an organism, there is a possibility of pleiotropic effects, where the introduced gene interact with the plant native genes in unexpected ways, potentially producing toxic substance. In that, the antinutrients. Some GM crops may produce higher level of antinutrients, which are compounds that interfere with the body's absorption of essential nutrients like vitamins and minerals. This could lead to nutrient deficiencies in the long terms. Okay, then next one is the cancer. There may be cancer risk concern, the DNA alteration and carcinogenesis. One of the most controversial claims is that GM food could increase the risk of cancer. The reasoning behind this concern is the inserting foreign DNA into crops could potentially cause unintended mutation in the plants. These genetic changes might increase the risk of cancer in humans consuming these products. Additionally, concerns have been raised about certain chemicals used in conjunction with GM crops such as glyphosate, a herbicide used with the herbicide-resistant crop so which some studies have linked to the cancer. Then another one is the antibiotic resistance. There is another concern for the antibiotic resistance genes in GM food. Some GM crops have been engineered using genes that confer resistance to antibiotic as a marker to identify successful genetic modification. There is a concern that these antibiotic resistance genes could transfer to bacteria in the human gut, leading to the development of antibiotic resistance infection. If bacteria in the human body acquire this resistance gene, it could reduce the effectiveness of antibiotic treatments, contributing to growing global issues of antibiotic resistance. The effect on ecosystem and the biodiversity. The potential environmental impacts is the alteration of ecosystem dynamics, reduced biodiversity, and the impact on soil health and microbes. So, in that, the GM crops could disrupt the natural ecosystem by introducing traits that altered interaction between plants, animals, and microorganisms. Then, reduce biodiversity. Genetically modified crops can reduce the biodiversity by encouraging monoculture when large areas are planted with a single type of crop, reducing the variety of species in that ecosystem. And the impact on soil health and microbiomes. Genetically modified crops can affect soil ecosystem, including soil microbiomes, which play a crucial role in nutrients cycling and the plant health. Herbicide-resistant crops, for example, could lead to overuse of herbicide, negatively affect the soil organism. The last one is the hazards of GM food. There is the key concerns. One is the absorption of introduced gene, in that there is a concern that gene introduced into the genetically modified plants could be absorbed by human body through digestion, although the likelihood of this occurring is extremely low. Then the transfer of antibiotic resistance gene. If the antibiotic resistance gene are present in genetically modified food, they could potentially transfer to gut bacteria, contributing to the rise of antibiotic-resistant infections. Then next is the pleiotropic and insertional effects. Genetic modification can use pleiotropic effects, where one genetic change has multiple unintended consequences. Similarly, insertional effect can occur when inserted gene disrupt other important genes. Then next is the anti-nutrient increase, in that genetically modified crop may increase level of anti-nutrients, substance that reduce the nutrient absorption, leading to the possible negative health effect. And last one is the viral DNA use. In this, some genetically modified crops are modified using viral DNA, raising concern that these viral components could have unintended effects on human health, such as triggering disease. In this, focuses on the global adoption of genetically modified crops and the regulatory framework governing them. In that, the global adoption, genetically modified crops and agricultural land, the GM crops currently cover 3.7% of global agricultural land. However, vast the majority 98% of crops are grown in just 10 countries. The global genetically modified crops production, in that these countries are responsible for 99% of all genetically modified crops production worldwide, indicating that the genetically modified technology is concentrated in few regions despite its global importance. Then the the genetically modified crops are mostly grown in the US, Brazil, etc. So the regulatory frameworks, several key organizations and protocols play a role in regulating genetically modified crops and foods to ensure the safety, ethical use, and compliance with the international standard. This includes the FDA, Food and Drug Administration, oversees the safety of genetically modified foods and drugs in the United States, ensuring they are safe for the consumption. Then USDA, its United States Department of Agriculture, regulates the GM crops, agriculture impacts, and including the environmental concern like pest resistance. The next is the EPA, Environmental Protection Agency, ensures that genetically modified crops, especially those modified to resist pests, don't harm the environment. And next one is the EFSA, European Food Safety Authority. This is responsible for assessing the safety of genetically modified crops and foods in the European Union, providing the scientific advice. Then last one is the Cartagena Protocol, an international agreement under the Convention on Biological Diversity that regulates the safe transfer, handling, and the use of genetic organisms across the border. Then another is the Codex Alimentarius, a set of international food standards that includes the guidelines for genetic modified foods to ensure they are safe and labeled correctly. The key case studies and controversies that have been discussed in the scientific literature concerning genetically modified foods. First one is the genetically modified food, that safety, risk, and public concern, a review. This review paper covers the major safety, environmental, and health risks associated with GM food. It also addresses the public concern about consuming genetically modified foods and evaluate whether these concerns are scientifically justified. It points out the challenges of balancing biotechnology with consumer acceptance and safety. Then, second one is the controversy of genetically modified food. In this article, explore the wider debate surrounding the genetically modified food. It highlights how media, public perception, and lack of knowledge have fueled controversies. The biggest fear include potential health impacts such as cancer, allergic reaction, and environmental issues like biodiversity loss and contamination of non-GM crops. This study underscores the importance of communicating the scientific evidence clearly to resolve this concern. Then third one is the health risk of genetically modified food. This case study specially focuses on the potential health risks posed by the genetically modified food, such as allergic reaction, toxicity, and other long-term effects. It reviews the scientific research on how GM foods might affect the human health, especially with their impact on digestion and metabolism. This article adds to the ongoing debates by examining a real world cases of health risk linked to the genetically modified foods. Together, these studies represent the scientific discourse of GM food. They address the public health risk, environmental concern, and the need for transparent communication between the scientists and consumers to mitigate fears and ensure the safe adoption of the genetically modified technology. The future directions and research areas for genetically modified organisms, emphasizing the need for innovation, safety, and public trust as we move forward with this technology. The first one is the innovation in GMO application with vigilant oversight. As the GM technology evolves, we will likely see more innovative applications such as GM crop designed to tackle climate change by resisting droughts, improving nutrient content, or reducing the need for chemical inputs like pesticides. However, with innovation must come vigilant oversight. Regulatory bodies have scientists will need to constantly evaluate the safety of this advancement to prevent any unintended consequences for human health or the environment. And second is the enhanced safety testing and risk assessment. The future of GMO requires more comprehensive safety testing. Current testing focuses on short-term impacts, but as the technology evolves, there will be a growing need for long-term studies. These include assessing the potential for new allergens, toxins, and unintended genetic effects that may arise. By enhancing the risk assessment protocols, regulatory agencies can better protect consumers and ensures that new GMO are safe for widespread use. Then third is the strengthening the public trust through the transparency. Public trust is a significant issue with GM food. As we continue to develop the use of GMO, it is crucial to ensure that there is a transparency at every level, from the lab to the market. This means clear labeling, sharing safety test results, and making information accessible to the public. By being open about the research and development process, we can help elevate public concern and build trust in the technology. Then last one is the public perception and the ethical consideration. Finally, public perceptions and ethical concern will shape the future of GM food. The public often question whether GMO are natural, safe, and ethically sound. Addressing these issues requires not only scientific evidence but also engagement with social values. Ongoing research will need to consider how GMO align with the consumer ethics, environmental sustainability, and global food security. Engaging in conversation about these topics will be essential in shaping future policy and the consumer acceptance. So, in the conclusion, we understand the complex consumer attitude for the public opinion on the GM food is diverse, with some consumers accepting them while others remain resistant. Then the public trust. It is the more essential to rebuild the public trust in the regulatory bodies and the safety of the GM food. Then third one is the transparent regulation, the need of transparent regulatory framework that involves the public in the decision-making processes. Then the public involvement. There is also engagement of the public in discussions about the GM foods and involving them in the regulatory processes can increase the acceptance and the confidence for the genetically modified food and the crops. Thank you. So that brings us to the end of the presentation. Thank you all for your time and attention. I hope this session has provided valuable insights into genetically modified foods, their potential impacts, and the future direction for research in this field. Thank you once again.